All right, here we go with part two on viruses. So we're going to talk a little bit more about what viruses have to achieve if they're going to reproduce once they enter into a host cell. And so how do viruses know who their host cells are? Which ones are they going to go into? Now there's more than one thing that can determine that, but usually one of the key things is that they have to bind to a host cell receptor. And we've talked about that concept before. We talked about that with bacteria. They have little many pathogenic bacteria have fimbriae spikes that help them attach to certain cells on certain tissues inside the body. Same deal for viruses. So for example, here's an HIV virus. HIV has these glycoprotein spikes that protrude from the surface. Those are called GP120. That stands for glycoprotein. And the 120 refers to the molecular weight of that glycoprotein. It's 120 kilodaltons, or that means 1,000 daltons. That's a measurement for molecular mass that you learn about in chemistry. That spike specifically binds to a molecule called CD4. That's also a kind of glycoprotein, and that is located on the surfaces of some of our T lymphocytes. And it's also located on macrophages and a few other cell types in the body. Like if you may remember learning a little bit in biology 201 about the microglia in your brain and spinal cord and the central nervous system. Those are like macrophages that live permanently in your brain and spinal cords. And HIV can infect those cells as well. But HIV is not going to infect cells in your body unless they have this molecule called CD4 on the surface, and not many cell types in the body have that. But CD4 and GP120 come together, you know, kind of fit. All right, sorry about that. I had a brief interruption there for a second. Okay, so as I was mentioning, in the case of HIV, it has these glycoproteins on its surface called GP120, and they very specifically bind to a protein called CD4 that is found only on the surfaces of certain cell types in the human body. And those cells are involved with our immune system, and that's why HIV cripples the immune system after a number of years of infection. So all viruses have to bind to some sort of cell surface receptor, and they're only going to attach to cells that have those receptors. That's going to be one of the key things that determines where a virus goes in the body. Some viruses, their receptors are located on many different cell types in the human body, whereas others are a lot more restricted, kind of like HIV as far as where they will go. Other things can play a role as well. It's not only the receptor that determines where a virus is going to go inside the human body. Another example is temperature. The common cold virus, rhinovirus, the reason why it really only replicates up in the upper respiratory tract, like in your nasal cavity and in your pharynx and so forth, is because the temperatures in those areas are a little bit cooler than normal body temperature, because we're constantly breathing in cooler air that's below 98.6 Fahrenheit. And the common cold virus prefers to replicate at temperatures that are a little below human body temperature. The receptor for human rhinovirus is actually present on many different cell types in the body, but the virus just does not replicate very well in other locations. There are other things that can play roles as well inside the cells. There are factors inside cells that help viruses replicate and assemble themselves, and some cells can assist viruses with those types of things, and others cannot. So it does go beyond there. All right, the viral replication cycle. What we're going to do, I'm going to generically talk about how viruses replicate, and then later we're going to see some more specific example viruses and some of their little quirks and nuances as far as how they actually wind up making new copies. All viruses have to go through these 5 steps in order to replicate themselves. The first one we were just talking about, that's attachment. So the virus over here, number 1 in this diagram from your textbook, the virus is going to have to attach to its receptor on the surface of the cell. Then you're going to have entry. And viruses differ in how they enter. In some cases, the whole virus itself will come into the cytoplasm of the cell, and then it'll break apart, and the genome will come out. That little yellow squiggly thing there is supposed to represent an RNA genome of an RNA virus. In other cases, the virus just sits there at the surface and squirts the genome on the inside. So there are differences. The key thing, though, is you got to get that genome inside the cytoplasm of the host cell, because the genome has the genes of the virus. Those are the genetic recipes that have to be read, that have to be expressed if we're going to make new viruses. So once we've entered, we synthesize. So you got to make more viral genomes like you see over here, and then you've got to, the host cell uses its ribosomes to make the virus's proteins, because you got to make new capsids. And if the virus has spikes sticking out from the surface, you got to make new spikes. All those things have to be synthesized. Viruses do not have their own ribosomes. That's one of the reasons why they've got to use a host cell in order to replicate themselves. So that's one of the key things. They're hijacking the ribosomes of the host cell in order to make their proteins. All right, and once you've made all of these new viral parts, you assemble them together. And then finally, you got to get these viruses out of the host cell. That's called release. In some cases, the host cell will actually break apart. That's called lysis. We've heard that word before. The host cell will break apart and die. In other cases, the virus does something called budding, which we'll take a closer look at later. But what budding means is you'll have the genome of the virus surrounded by a capsid, like you see right there, and that capsid enclosed genome pushes itself through a cell membrane and wraps itself with an envelope. Remember from the last video clip, we said an envelope is actually some of the phospholipid membrane of the host cell that the virus has stolen as it assembles itself and buds from the host cell. So those are the 5 key phases you've got to go through in order to create new viruses inside the host cell. Notice this word here too, virion. Virion is a term that refers to a virus particle. You'll also see infectious virion. An infectious virion is a single virus particle that is fully mature and is capable of infecting a new host cell. You can have immature virions as well. That means maybe you've assembled a particle, but it hasn't quite configured itself perfectly yet in order to be able to infect a new host. So how about some, I mentioned that different viruses use different strategies in order to enter their host cells. And this diagram, which is actually from an older textbook, is just showing you some differences, lots of variety with viruses. One method is called direct penetration of the genome. And here, this happens with non-enveloped viruses, so they just have a capsid shell like you see here, genome on the inside. The virus attaches to its host cell receptor, but the virus shell does not actually enter the host. It just squirts the genome inside the cytoplasm of the host cell. And then the host cell is going to see this genome and start reading those genetic recipes and making new viruses. Another method called membrane fusion, if you have an enveloped virus, remember that envelope actually consists of a phospholipid bilayer. And so once that virus attaches to a receptor on the host cell, the phospholipid bilayer of the virus's envelope can actually melt into the host cell's plasma membrane. They can fuse together because all those phospholipids, they love each other. So they just kind of naturally fuse together, and the capsid shell on the inside can be released into the host cell, and then eventually what you need to have happen is that capsid needs to break apart. It needs to open up and let the genome out. That's called membrane fusion. And then finally, a lot of viruses hijack a natural cellular process, endocytosis, where something attaches to the surface of a host cell and then it gets engulfed inside a membrane covered vesicle and enters the host cell that way. That's a natural eukaryotic cell process you learn about when you're learning about how things move across cell membranes. And that's what this virus you see over here is doing. This can happen with some non-envelope viruses, and some envelope viruses do this. But the virus attaches to the cell surface, and then the host cell membrane, a part of it pinches off and wraps the virus inside a little compartment called a vesicle. And then somehow the capsid has to come out of that vesicle, or the genome. The capsid can break apart and the genome can come out as well. So those are just some different ways that viruses can use to get into their host cells. And this one is especially kind of interesting because it really is hijacking a natural cellular process. It's tricking its host cell into thinking, hey, this is something we need to engulf and bring inside the cell because this is going to be something useful or beneficial. And it turns out it's a virus that's coming in to hijack the cell's machinery to replicate itself. Interestingly, when it comes to coming up with new types of, we don't have a lot of drugs to treat viral infections, but one of the steps during the life cycle that people are really looking at is this one. If you can block attachment, or you can block membrane fusion, you can block the release of the genome. Can you prevent the capsid from opening up and releasing the genome inside the host cell? Those would all be good strategies for helping us maybe combat certain types of viruses. All right, a little bit more about how viruses replicate inside their host cells. So I mentioned when we went over the different types of viruses, we said that there were RNA viruses and there are DNA viruses. And among your RNA viruses, we said there are positive single-strand RNA viruses, and there are negative single-strand RNA viruses. And this is a figure from your textbook. A positive strand RNA virus, or a positive or plus strand RNA virus, you'll hear different terms for that. What that means is once that genome enters the host cell sees that as a messenger RNA. It thinks it's a messenger RNA. So it is going to do translation. This is a viral protein, this green squiggle you see over here. And viruses will typically make more than one protein, but they're just showing you there one to keep the diagram more simplified. So you're making those virus proteins, and then also you've got to make more genomes. In order to make more genomes, what type of enzyme did we say makes RNA? RNA polymerases. So viruses will have genes that code for their own RNA polymerases. RNA polymerases are proteins. So you're going to have to make those RNA polymerases of the virus. The RNA polymerase is going to take these genomes and make a complementary copy. Those are called negative RNA strands, or negative sense RNA. So that means if you got an A here, it's going to put a U over here, a C over here, it's going to put a G. So it's going to make a copy. And then that RNA polymerase reads these guys and does the reverse. So it'll make more plus strand RNA viruses over here. So you go from plus to minus to plus, and that's done through complementary base pairing, just like we've talked about before. Then those new genomes get packaged inside the virus's capsid proteins, and you assemble the new viruses, and you're all done. Negative strand RNA viruses are different. So when their RNAs get released inside the host cell does not see those as messenger RNAs. You first have to take the virus's RNA polymerase enzyme and make a complementary copy of this. You've got to make a plus strand, because that's the type that the cell will see as a messenger RNA. And once you've done that, those can be read, those can be translated to make the virus's proteins. And then others will be used as templates to make more minus strand RNAs. So you have to do RNA replication as well. You go from minus to plus to minus using that enzyme. Once you have more of these minus strand RNA genomes and all the viral proteins you need, you assemble together and make your your new viruses like you see over here. So this is something that's very different from living things. As far as we know, there are no cells on the planet that have RNA genomes and never have DNA as part of their life cycle. These viruses don't use DNA at all. They strictly use RNA. Then there are some viruses that are called double-stranded RNA viruses, where they come into the host cell with both a plus and a negative sense RNA, and they are base paired with each other, kind of like what you have with DNA. And you need to break those apart because the pluses are going to be your messenger RNAs that are going to be translated to make your proteins. And then you've also got to do RNA replication, because you got to make more of those double-stranded RNAs, and then you assemble all of those things together and make new virus particles. Then there's a special type of virus called a retrovirus. HIV is a famous example of a retrovirus. So in the virus particle, you have a positive strand RNA, and that actually gets converted to a single-stranded DNA that's complementary to this. So if you have an A here, you're going to have a T over here, it's DNA. G over here, it'll be C over here, etc. And then that gets read, and you make a complementary copy, and now you have a double-stranded DNA genome. So you started with RNA and went this way and made a double-stranded DNA. So that is the opposite of what happens in our cells when we're thinking about transcription, and that process has a special name, and that is called reverse transcription. If my pen will write here, transcription. Change pen colors there. That is a special process where you go from RNA to double-stranded DNA. The reverse of transcription, where you're going from double-stranded DNA to RNA. And that is done by viruses called retroviruses, and HIV is a famous example. And there's an enzyme that's used to accomplish that. That enzyme is called reverse transcriptase. And our cells don't need reverse transcriptase for anything. Reverse transcriptase is an HIV enzyme. And so a number of the drugs that are given to AIDS patients to help keep the virus under control are called reverse transcriptase inhibitors, because they're drugs that shut down the function of the reverse transcriptase enzymes that virus needs to replicate its genome. And then notice over here as well, all right, so you get that double-stranded DNA copy of the genome. The virus itself needs more of these plus-strand RNAs inside the new viral particles. So you'll do regular transcription. You'll go from double-stranded DNA to RNA to make new genomes that are going to wind up in the retrovirus particles. And again, HIV is an example. And those messenger RNAs will also be translated to make the virus's proteins that it needs as well. All right, among your DNA viruses, they tend to behave more like our cells do. Double-stranded DNA viruses have double-stranded DNA genomes. So just like our cells' genomes undergo DNA replication, they'll do that to make new genomes. The genes of the virus on here get transcribed. You do transcription to make messenger RNAs, and then you do translation to make the virus's proteins. Then there are some DNA viruses that are single-stranded, and they're also called positive strand DNA viruses as well. And during their replication cycles, this gets converted to a double-stranded DNA, and then that is used to make more single strands, because those are the ones you've got to put into the new virus particles. And this gets treated like some of our own DNA. You do transcription of the genes, and then you do translation to make the viruses proteins. So not all viruses, some viruses behave very much like our genomes do. Their genomes behave very much like ours do. But then you've got these other types of viruses that do things that violate all of the rules. That central dogma of going from DNA to RNA to protein does not always hold up when it comes to viruses. All right, so the viral genome, what do you have on there? You've got a series of genes. Different viruses will have a different number of genes. It depends on how complex they are, how many different kinds of proteins they need to make. But those proteins include your structural proteins. Those are your capsid proteins that make up the shell, the glycoproteins that sit in the envelope, those little spikes that protrude from the surface. Sometimes you have filler proteins in between called matrix proteins as well. And then other types of proteins of the virus are called replication proteins. Many of those are enzymes that the virus uses to make new copies of its genome, like the reverse transcriptase enzyme of HIV we were just talking about. Or with your RNA viruses, they will make their own RNA polymerase enzymes that do things like go from plus strand RNA to minus and then back to new plus strands, or from negative to plus to negative, depending on the type of virus. And then there will be others as well. It's not quite that simple. Many viruses have a number of other kinds of proteins that they need in order to fulfill their life cycles. And again, thinking about drugs, if we want to come up with good antiviral drugs, we still don't have a lot of those out there that are available. You got to come up with things that are not going to harm the host cell. Hey, it's great if you come up with some drug that interrupts the replication of a virus, but if it also harms the host cell, it's not going to be a good drug. So, and that's been the challenge. We need to come up with drugs that interfere with unique things the virus does, like the reverse transcriptase of HIV, or if we could come up with drugs that would screw up a virus's RNA polymerase enzymes, but it wouldn't screw up our RNA polymerase enzymes, that would be another good target. Can you block the virus from attaching to its receptor? Can you prevent it from uncoding itself after it attaches to a host cell? Any of those types of things would be good. Tamiflu, which is one of those few antiviral drugs that we do have out there that quite a few people take early after flu infection. Interestingly, what it does, if we've got a host cell here, flu viruses bud from the surface. These are some new flu viruses coming out of their host cell. Well, those flu viruses need to detach. The problem is the host cells have flu receptors in them in the surface. So when a new flu virus buds out of its host, those new viruses have a tendency to just reattach to their host cell receptors. It's really easy for them to do that. So there's actually an enzyme in the surface of the flu viruses that helps clip the new flu virus particles off so they can be released to the outside. So Tamiflu inhibits that enzyme that clips the new flu viruses off of their host cell. So because of that, your new flu viruses just wind up being stuck on their original host cell and they can't spread elsewhere in the body. So that winds up helping to limit the spread of those viruses elsewhere. All right, and we did mention budding is a process by which enveloped viruses exit their host cells. This is a diagram from your textbook. Now over here they're actually showing you a colorized electron micrograph of a real virus pushing itself out of a host cell that you see here. So the yellowish colored material in the center is going to be the viral genome, and you can see some of the other layers around it. Like there's one right there that is not quite fully pinched itself off of the host cell membrane. Here's another one over here, and here's one that's just kind of starting the process of pushing itself through. And now over here on this diagram, they're showing you we've got an RNA virus that's going to bud, and it's floating around. The RNA genome is here in the cytoplasm. the capsid proteins, they're in those little purple specks, assemble themselves around the genome to protect it. And then sitting up here in the host cell membrane, you have another viral protein called the matrix. And then these little green spikes here are glycoproteins of the virus. And the capsid with the genome on the inside is attracted to these other viral proteins that are already poised and waiting there in the membrane like a magnet. I mean, they come together very strongly, and as that happens, the capsid winds up, the envelope with those proteins winds up wrapping itself around the capsid with the genome on the inside, and your new virus pinches itself off. That's called budding. All right, so I'll stop that video clip there. On the next one, I'm just going to go through and talk about a few highlights of some example viruses. There are many different, what we've talked about are some basic strategies that all or many viruses have to use to replicate themselves, but every virus has its own little quirks, and we're going to discuss a few examples of those quirks for some specific viruses that we hear quite a bit about.